Okay, so we will get started. Hello, everyone. Thank you so much for coming today to our second week of the Catalysis webinar. I'm really looking forward to introducing our speaker, Dr. Christopher. To introduce myself, I'm Lorianne Schultz, and we also have on here Katie Slavinska, and we're both from the American Vacuum Society at the University of Central Florida. The way that this webinar is formatted is after Dr. Christopher gives his 30 to 45-minute talk, then there will be a question and answer period. So if you have questions anytime during the talk, just type them into the chat. Don't try to unmute yourself or anything like that, please. Just type them. And then at the end of the presentation, I'll read those questions out loud to Dr. Christopher, and we'll get to as many as we can during that period. So as I said, I'm really looking forward to introducing our speaker, Dr. Christopher. He obtained his BS in chemical engineering from the University of California, Santa Barbara in 2006, and his PhD in chemical engineering at the University of Michigan in 2011. Starting in 2011, Dr. Christopher worked as an assistant professor at UC Riverside until he moved his group to UC Santa Barbara in 2017 and became the Mellichamp Chair of Sustainable Manufacturing. He has been serving as a senior editor of ACS Energy Letters from 2016. Dr. Christopher and his group's work in the field of catalysis has been recognized with many awards, including the AIChE Catalysis and Reaction Engineering Division's Young Investigator Award from ACS, the Presidential Early Career Award for Scientists and Engineers from the U.S. Army Research Office, the Early Career Award from the NSF, the U.S. Army Research Office's Young Investigator Award, and the Young Scientist Award from the International Congress of Catalysis. And today, Dr. Christopher will share a lecture on atomically dispersed platinum group metal catalysts, distinct reactivity, uniformity, structural evolution, and pathways to increased functionality. So I'll pass this on to you, Dr. Christopher, for whenever you're ready.
All right, let me see if I can. Can you see that well?
That looks great, thank you.
Cool. So, Lorianne, thank you very much for a nice introduction, as well as for the invitation to come present here. This is kind of an interesting format, and I'm excited to see how it goes. I guess I've never really given a seminar to a group of people across the world that I haven't met before, so thanks for the opportunity. So today I'm going to be discussing some work we've done over the past 4 or 5 years in my group, looking at atomically dispersed platinum group metal catalysts. I can tell you on the front end that I've never given this talk in this way, so I don't know why I decided to put something different together for today, but I did. So we'll see how it goes. Let me see if I can go backwards. Sorry. And also, as you all know, we're all kind of stuck at home mostly, and so I apologize if anybody else makes a cameo in my talk today, whether that's one of my kids or my cat or the gardener or something like this. So please just bear with me. So this is work done over the past 5 or so years by various PhD students and postdocs in my group. And we've also collaborated a lot with Xiaoxing Pan's group at University of California in Irvine on microscopy, Simon Baer's group up at the Stanford Synchrotron Radiation Lab doing X-ray absorption spectroscopy, and then Gianfranco Paccioni doing theoretical calculations, as well as a friend of mine, Bean Getsoyan at Ford Motor Company. So let's get started. So I don't think this is a necessary slide for a group like this, but I just thought I would throw it in here to give some of the motivation for the work that we do. So broadly, heterogeneous catalysis, I would say, is one of the most enabling and important technologies that enables kind of the way that we live now. The primary goal of catalytic processes is converting natural resources, so things like oil that we dig up, natural gas that we pull out of the ground, even nitrogen that we extract from the air, and converting these to higher valued chemicals, whether that's the gasoline we use to drive our cars or the fertilizer we use to grow plants and food and crops, or whether that's plastic bottles that we get from the ethylene that comes from either natural gas cracking or from oil. All of this is driven by some catalytic process. So some of the numbers that are put out there is that catalysis contributes in one way or another to something like 30% of the world's gross domestic product. So I think this year the world's gross domestic product is somewhere around the order of, I think it's $70 trillion. So you do the math, that's a huge amount of money. And on the flip side, catalytic converters are also, catalysis is also what enables us to have such high levels of gross domestic product while minimizing the pollution. So catalytic converters are the primary technology we use to make the burning of gasoline reasonably minimally impact our environment, but there's lots of other processes as well. So catalysis in general is a hugely impactful area and something that will enable us to further grow in the way that we all lived and hopefully try to do so in a sustainable way. So the specific class of materials that I'll talk about today are supported metal catalysts. So specifically, these are used broadly in many of the reactions I just described on the previous page. And when you think about a supported metal catalyst, you can think about it in lots of different ways. But the easiest way for me to think about it is technologically, when something is put into a reactor as a supported metal catalyst, you usually have a domain that looks like this. That's some sort of oxide material, most typically. Usually these are thermally stable oxides like silica, alumina, sometimes things like titania, sometimes zeolites, which are silica aluminates. And these are often thermally stable, mechanically stable, and extremely high surface area materials. So oftentimes somewhere between 100 or 1,000 meters squared of internal surface area per gram of material. And the reason you want a really high surface area material is because you want to be able to load this with a lot of nanometer scale active metal species. So within a catalyst, you often have a support, which on a 0th order approximation is simply the carrier for then your active species, which is usually on the scale of some nanometer size metal or metal oxide particle. So this cartoon, you can see maybe a little better depiction of it over here. This is a classic SBA 15 material. So you see mesopores going through this. This is a silica. And then these dark spots in here in this TEM micrograph are metal particles, I think platinum in this case, loaded inside of this system. So if we think about what makes up supported metal catalysts, you can think about it at a little bit finer scale. So here my red and my white balls are an oxide support. And these various different domains here are active metal species. And so I drew these a few different ways. In this case, at the smallest scale, this is something like a 1 nanometer particle, metal particle. This is maybe up to a 2 or 3 nanometer diameter metal particle. The blue balls here are surface atoms that are well coordinated to their neighbors. So they're making something like 8 or 9 bonds to their neighbors. These green ones are edges, and these are making maybe 5 or 6 or 7 bonds to their neighbors. And you can imagine that their varying coordination causes very different reactivities. If you take this even a step further, you can imagine these little yellow ones down here where you have single precious metal atoms dispersed on a support. Now, in this case, the local coordination environment is extremely different than you would find in a larger metal particle. So you're bonding to oxygens or metals on the support rather than neighboring metal atoms. And so as a result, you could imagine that the oxidation state of this species may be different than these. The coordination environment would tell you that the reactivity should be different. And of course, if you have a reaction where you need communication of multiple sites, this will happen very differently on this sort of site than it does on this sort of site. So with all of that in mind, this also has an advantage when you can shrink these particles down to this level, that you're perfectly utilizing expensive and precious metals. So from some sustainability of precious metal utilization approach or perspective, being able to shrink these down and perfectly use all of our metals so that every metal atom I put into a catalyst is exposed and able to do chemistry is a really advantageous potential property. The question, which is motivated by what I just discussed, though, is clearly the coordination environment of this species is different than these ones. So is the reactivity different? It clearly should be distinct. Would it be beneficial? These are the sorts of questions that we're trying to ask. So I think it's always important to look at any project that you're working on and try to understand the history of these things. Now, these sorts of catalysts, which some people are calling single atom catalysts, where you have these single precious metal atoms dispersed on oxides, have received a lot of attention recently, but I think it's worth remembering that there's a lot of history around these materials. So we'll briefly go through some of that. So there's this beautiful paper from John Yates's group from 1979 or so, looking at the infrared spectra of chemisorbed CO on rhodium. And in it, there's a passage. And the passage here I highlight said, for alumina-supported rhodium, Yang and Garland first postulated that at low rhodium concentrations, where sintering did not occur at reduction temperatures below 200 C, rhodium existed in a condition closely approaching atomic dispersion, i.e. isolated rhodium atoms. This picture has been confirmed by others, although there are differences in interpretation regarding the degree of dispersion. So a couple things to point out here. First, the paper which he was discussing, which was from his advisor, John Yates's advisor, actually came out in 1957. So for over, I don't know how many years that is, 70 years, 60, 70 years, people have been thinking about the reactivity of atomically dispersed or single metal atoms on oxides. And you can see even at this point in 79 or so, there was still some arguments on interpretation on whether or not the rhodium was in small rafts or single atoms or how this looked. This paper specifically had very nice infrared spectra in here, and I'll talk a little bit more about this in a few slides, but when you adsorb CO to a catalyst where rhodium has been dispersed on an oxide, you see all sorts of different stretching bands. This should tell you that there's lots of different coordination environments that CO is binding to. And it's interesting to try to understand what those coordination environments are. So in this paper, Yates nicely tried to identify a few different coordination environments, those that can happen only on single atoms and those that can happen only on clusters. So this is just to give you perspective that there's a lot of information out there about this class of materials, and it's not just a new field. There's actually a lot that's been done previously. Second, some motivation. So obviously when you're trying to figure out whether or not a class of materials is worth studying in lots of different detail, you would want to convince yourself that there's some interesting properties to motivate detailed studies. And so I highlighted just one or two studies from some time ago showing interesting reactivity of atomically dispersed metal catalysts. So this is a classic paper from Bruce Gates's group. What they looked at is they looked at mononuclear rhenium species on an MgO support. These were carbonyl species. And then they compared these mononuclear structures to a trinuclear species, so 3 rhenium atoms at the same time. Effectively, what they found is if you feed cyclopropane to these catalysts, whether you have 1 atom or 3 atoms in your active sites, cyclopropane will isomerize to propene, and you can do some hydrogenation to make propane. This happens on both types of sites. However, only when you have those 3 atoms together in an active site, so a rhenium, a trinuclear rhenium species, do you get this hydrogenolysis reaction where you start breaking carbon-carbon bonds, producing methane, producing ethane. So this was demonstrating structure sensitivity down to the limit of 1 atom versus 3 atoms in the active site, showing that there may be some interesting behavior if you can do catalysis by species that are just 1 atom active sites. You can take this further in another paper. I tried to pick some surface science literature as well, just to show that this has been done in many different fields. This is a paper from Uli Heitz's group in 2000 or so, where they were looking at size-selected clustered palladium deposition onto MgO. They were doing acetylene TPDs from this system. And what you could see is at low temperature, acetylene would cyclotrimerize into benzene, even on 1 palladium atom on MgO. So tells us that, I think the title of this paper is 1 atom is enough, that these sorts of structures where you can shrink active metal active sites down to the limit of 1 single metal atom may have interesting selectivity, the previous slide, and also can do relatively complex chemistries. Take this a step further, and now 10 years later, you can see each one of my examples were about 10 years apart or so. About 10 years later, and this was the paper from Tao Zhang's group that I think sparked a reemergence of interest in this area of so-called single atom catalysts or atomically dispersed catalysts. And here Tao Zhang's group was looking at platinum atoms on iron oxide. And they compared 2 samples. One sample where they said, we only have single platinum atoms showing up as these bright spots, and I'll talk about that more in a TEM image on an iron oxide lattice. And in a sample B, you have some small clusters here. If you went and measured the CO oxidation rate on these different catalysts, what you would find is the one that only had single atoms had a bit higher turnover frequency than the one that had clusters in it. And so from this, they inferred that these single atoms can have higher reactivity. So in the 3 examples we've seen, you have these dispersed species that can have a higher reactivity, can do complex chemistries, and can potentially have different selectivities than, say, small clusters of the same metal. So this case, I think, sparked interest in this area primarily because advancements in aberration-corrected electron microscopy allowed you to actually see these single atoms instead of just having to infer their existence from characterization or interpretation of spectroscopy. So this leads us to the question of, there's long been interest in these materials. What has changed? So in my perspective, advances in transmission electron microscopy, which allow us to actually see these species, and the coupling of this with spectroscopy and theory allow us to actually model these active sites in a fair amount of detail, and I'll show some examples of that today. Second is there's continued increase in focus on atom-efficient chemical conversion. So can we make heterogeneous catalysts that rival organometallic complexes in their uniformity and selectivity, such that we can take advantage of the operational capabilities of a heterogeneous catalyst? You have a stationary phase, so separation processes are not a challenge or as much of a challenge, while still being able to impart these high levels of selectivity. And this class of material begins to think about how we can go that direction. So there is evidence of distinct reactivity I just went over. So what more can we do here? What are the interesting things in my perspective? So from my perspective, there's always the question in catalysis of whether a subpopulation of clusters could be the active site. So in each of the previous examples I gave you, there was no analysis in each sample of whether or not there could have been a subpopulation of other active sites that was actually doing the chemistry. There's really difficulty in characterizing and understanding exactly what the active site coordination environments look like and whether or not they're fluxional under reaction conditions. And finally, there's questions on whether we can engineer the local coordination of these species to control their reactivity. And so this is kind of what an overview, what I'll talk about today. So the overview of the talk, I'll first talk about some distinct reactivity and how we develop these relationships trying to use quantitative approaches rather than just comparing two samples that have some characteristics of different active sites. Next, we'll talk about the uniformity of these single atom species. I'll go further in how the uniformity or how the coordination environment of these single atoms on an oxide can move and change. And then I'll end with some thoughts on how we can increase the complexity of these active sites and hopefully increase their functionality. So first question is, how do we develop rigorous structure-function relationships where we can convince ourselves that these atomically dispersed metals actually have the behavior we're assigning to them? So one approach, and this is not meant in any way to downput this particular paper, this is just an example where something like this was done because I do think this was pioneering work. So one case, you can look at two samples. You can look at electron microscopy and you can say, we could only see single atoms here, and here we could see lots of clusters. So let's compare their reactivities, and this will give us some measure of the difference in their performance. So we can make two materials and we can compare reactivity. There will always be a question of whether or not there was a subpopulation of species in this sample, which were predominantly associated with the measured reactivity. And there will be always a question of what the single atoms that you saw in this sample were doing and playing a role in the chemistry, and how do you differentiate those? It's very difficult to do from just looking at two different samples that from characterization approaches have some differences in structures. So a different approach that we've been trying to develop is instead trying to make a series of materials, maybe make 5, 6 different materials, and then develop approaches where we can try to quantify the relative fraction in each material of active sites exposed on nanoparticles or clusters, and active sites exposed on, say, single atoms. And if we do have these quantitative measurements of the relative fraction of each one of these sites, then we can go make a measurement of some intrinsic property of catalytic an intrinsic property of catalytic activity, something like a turnover frequency. And we could begin to correlate these quantitatively such that we can build quantitative or rigorous structure-function relationships. And I'll show some examples of how we've been trying to do this to really convince ourselves what the behavior of these active sites is. So the example I'll use for this is the catalytic CO2 reduction by hydrogen. There's a lot of interest in trying to reuse CO2 potentially as a feedstock for chemical processes. There's a lot of challenges with doing this, including actually capturing CO2 in significant quantities and concentrations that this is feasible. We were interested in understanding some of the fundamentals of the chemical conversions that can take place when you start reducing CO2 by hydrogen down. So if you do a simple 2-electron reduction, you can go to CO. The complete reduction sends you all the way to methane, and you can do some partial reduction to methanol, and all of these have various potential applications. Our interest was in rhodium catalysts, where it's known from a lot of different analyses that rhodium falls in the regime where you get primarily CH4 selectivity in this reaction. So on rhodium catalysts, there's maybe 100 or 1,000 papers in literature that show if you take CO2 and hydrogen, you react them on rhodium surfaces, you make mostly methane. There was a couple of reports, which I highlight here down at the bottom, that rhodium in certain ways, when you made it a certain way or pre-treated it a certain way or used a certain support, gave you high selectivity to CO, so a more mild reduction pathway, but there was really no insights into why. And our hypothesis was that maybe the catalysts that had been made here had relatively high populations of completely dispersed rhodium, and that those were responsible for production with CO, with the hypothesis being it may be challenging to do sequential hydrogenation processes on such a confined active site like a single rhodium atom. So what we did is we went to the literature and looked how we could make a series of materials where we thought we were varying the ratio of single atoms of rhodiums to clusters of rhodium within that material. And we found a nice paper, again from John Yates's group, who did a lot of work in this area, showing that if you simply take a nitrate rhodium precursor and do an impregnation process onto TiO2 supports, and you relatively mildly treat this with calcination and reduction processes, and you vary the weight loading from about 0.5% to 6%, that you can vary the relative populations of clusters to single atoms. And so we use this as a guide to say, let's make a series of materials and let's see if we can correlate the relative population of single atoms with the appearance of high selectivity to CO in this CO2 hydrogenation process. So we made a series of materials, and the first thing that we looked at is at the lowest weight loadings of rhodium and at the highest weight loadings of rhodium, we found relatively similar average rhodium particle sizes. So this was important to us because we knew as we were varying loading, maybe we would vary the relative fraction of these two species, but we might also change the nature of this one, making it larger, making it smaller, exposing more steps and edges or exposing more terraces, and having really two different ways we were influencing the potential chemistry. But the fact that the particle sizes were relatively similar across the whole weight loading allowed us to then just focus on whether or not a subpopulation of atomically dispersed or single atom rhodium species were causing differences in chemistry. So if you did aberration corrected TEM, and I'll show you much nicer images later, we definitely saw on all of our samples some clusters, some single atoms, telling us that we have some picture that might look something resembling this, but we needed a better way to quantify this. So the way that we went and tried to develop a quantification of these site distributions is the following. We used CO promolecule infrared spectroscopy. So this was kind of the most developed approach for studying dispersed rhodium species on oxides and trying to differentiate single atoms from clusters. So if I take, say, 2% of rhodium on TiO2, and that's what this spectra is for here, I oxidize the catalyst to remove ligands remaining from depositing my salt. I mildly reduce it in hydrogen at, say, 250 C, so that I take any oxidized rhodium clusters and I convert them to metallic rhodium clusters. And then I adsorb CO at saturation coverage at ambient temperature, or even below ambient temperature. And then I take an infrared spectra of this system, I get all these different beautiful bands. And these bands had been rigorously assigned by many people to a few different coordinating environments for CO. So if CO is linearly bonding to a rhodium atom on a rhodium cluster, you get a stretch around 2060 that shows up. If CO is bonding in a bridge fashion to a rhodium cluster, then what you get is this bridge bonded CO, which has a stretch down around 1850 or so. However, if CO bonds to isolated rhodium species or single rhodium atoms, actually what you form is a dicarbonyl structure. So on the other side of this, we should be making two bonds to the oxygen so that we have essentially a square planar complex. And this dicarbonyl can stretch in two different ways. You can have a symmetric, which is this stretch here, or an asymmetric stretch of the dicarbonyl. So through doing this infrared spectroscopy, we can decompose what exists, that it's exposed on the surface of a rhodium catalyst, into sites on nanoparticles by these two bands and sites on single atoms by these two bands. So we did this measurement for the series of materials that we had made from 6% of rhodium down to 0.5% of rhodium. And what you could see here is when you're at 0.5% of rhodium on TiO2, the primary bands are these two bands right here, which are associated with our gem dicarbonyl species, telling us we have mostly single atoms or atomically dispersed species. As we add more and more rhodium to the sample, this band associated with CO on linearly bound to rhodium particles, or this band associated with CO bridge bonded to rhodium particles grows, telling us that a higher fraction of the exposed sites are on rhodium particles. Now then what we can do is we can peak deconvolute these four different features. We can integrate their areas. We can normalize their areas by the relative extinction coefficients of each one of these stretches. And then we can further normalize by the number of COs to rhodium in each one of these bonding configurations. Here you have 2, here you have 1 per 2 rhodium atoms, and here you have 1 per 1. And through doing this, you can extract the relative fraction of exposed rhodium atoms existing as isolated species or nanoparticles. So we did this for that series of materials, and what you can see is as we increased rhodium weight loadings, the fraction of rhodium exposed as single atoms dropped from something like 60%, 20%, and down further as you got larger and larger loadings. And conversely, the fraction of sites exposed on nanoparticle surfaces went up. This is not surprising at all. That makes sense. You're putting more metal there. You should probably be having less and less single atoms because it's harder and harder to keep them apart. So we've made this series of materials. We quantified the fraction of sites existing in each type or each local environment, either on nanoparticles or isolated sites. So let's see if we can correlate this to their reactivity. So then what we can see is when we go measure selectivity here, and here we either had CO2 going to CO or CO2 going to methane, what you could see is that as we increased rhodium loading, we always saw higher and higher selectivity to methane. So this is consistent with the idea that methane formation happens over these rhodium nanoparticle sites. However, selectivity is not an intrinsic measurement that we can relate to site distribution, so we want to decompose selectivity into turnover frequencies. So what we did is we plotted the fraction of isolated rhodium atoms in each sample. So this is the fraction of exposed rhodium sites existing as isolated atoms as a function of weight loading, and that's the black line here. And then we compared this to, on this other axis, the turnover frequency for CO production. So just to remind you, that was this pathway here. And what you can see is there was a quantitative relationship under various different conditions. We tested this under a number of different reaction conditions, that there's a direct link between the fraction of sites exposed as isolated rhodium atoms and the turnover frequency for CO production we got out. Similarly, you can do the converse, where you'll relate the fraction of sites exposed on nanoparticle surfaces to the turnover frequency for methane. This is the other reaction that we could see happen. And once again, we have nice quantitative relationships. So this tells us together that we can quantitatively relate the turnover frequency for each product in this reaction to the two different types of sites we have. So the behavior of any given catalyst really is just dictated by the fraction of sites that you have that are isolated compared to the fraction of sites that you have that are nanoparticles. And this will give you the overall selectivity you have for a rhodium catalyst. And this way we could develop a structure function relationship. So this case I showed you was for CO2 reduction. We've done this many times recently for other chemistries using an almost identical approach. So in another case, we were looking at NO reduction by CO, which is how you reduce NOx in a three-way catalyst, a gasoline burning engine. One thing that we found here, which was interesting and surprising, is that single atoms in your catalytic converter are actually responsible for ammonia production. And that's because they're particularly good at doing the water gas shift reaction, which makes hydrogen in situ. And this can work to hydrogenate NO and produce ammonia. On the other hand, rhodium nanoparticles do the chemistry you would like to do, which is NO reacting with CO to form dinitrogen, which is the species we would most like to emit from the back of our car. So this told us that three-way catalysts should be made without atomically dispersed rhodium species. So this is to say these sorts of materials, single atom catalysts, these atomically dispersed metal catalysts are not great for everything. They have distinct selectivity here compared to clusters, but not in a way we would like. We've done this in other cases looking at commodity chemical production, so methanol carbonylation, which is done now using homogeneous catalysts in industry. We found that rhodium nanoparticles cannot be used to do this chemistry because they'll just decompose methanol into CO and hydrogen. Acidic sites on a support actually drive a dimerization of methanol reaction to make dimethyl ether, which is also not what we want to do when you're trying to do methanol carbonylation to make acetic acid. However, we found if you could make pairs of sites between isolated rhodium species and acid sites, that you could make some acetic acid. And so in this case, isolated rhodium species seem to offer some potential for heterogeneous methanol carbonylation. So I went over this just to tell you it's important to develop rigorous structure-function relationships. And we do that by relating the fraction of exposed sites on a given sample to some turnover frequency for a specific product. And doing this, we've been able to show, in many cases, single atoms and nanoparticles have very different reactivities for different chemistries. In some cases, it's potentially useful, so maybe we can do acetic acid production on heterogeneous catalysts rather than homogeneous catalysts based on this chemistry, or based on these sorts of active sites. But in some cases, it's not. So in the case of three-way catalysis, automotive catalytic converters, from what we see, if you put a bunch of single rhodium atoms in your catalytic converter, and if they actually survive through the tough process they have to go through, they would actually result in poorer performance of the system. So not always a benefit, certainly distinct, and the best way we can see to try to demonstrate their distinct reactivity is by quantitatively correlating the fraction of some site to the turnover frequency for some product over a series of materials. So the next, and that was my longest little section or part of this talk. Next, I'll talk a little bit about the uniformity of these active sites. So broadly, in the previous part of my talk, all I said is kind of all isolated sites or all single atoms had the same reactivity. But really, we like to question whether or not that's true. If you put a single metal atom on an oxide support, so I'm showing you here platinum on a titanium dioxide support, you could imagine many different coordination environments for this species to sit. It could make 2 bonds to oxygens, 3 bonds to oxygens, even more bonds to oxygens, and you can imagine each one of these would have different reactivity. You could also take the same structure, say a PTO2 structure, and put it on a terrace of an oxide, or put it on a step of an oxide, and again, it's gonna have different reactivity. And in the last case, you could imagine proximal hydroxyl groups having different reactivities or imparting some changes in reactivities to an atomically dispersed metal species. So the question we wanted to ask is whether or not we could take the first part of our talk where we said there is distinct reactivity, take it a few steps further and say, can we make materials that only exist in one of these coordination environments? And then can we identify what that coordination environment is? So the way that we've adopted trying to make uniform species is relatively simple. We use the electrostatic absorption of metal precursors onto oxide supports. We do this at extremely low loadings with the hypothesis that the lowest loadings will allow us to titrate the most energetic sites on the support with our metal precursor. And then we try to deposit about 1 metal atom per small oxide particle. So it's very challenging for them to hop together and form a cluster. So this is the general approach that we take. We also use, in this next section, I'll show a couple examples. We use a simply impregnation prepared catalyst where we just dry some salt onto some support and calcine and reduce it to form metal clusters as a comparison, just to assess uniformity. So how do we know when we've synthesized a uniform structure? So the first thing I always see is people wondering how you look at these TEM images and how you convince yourself that there's a single atom there. So what we do is we try to do line scans always, and we try to do line scans only when we've oriented our electron beam in the microscope along a particular zone axis of the support. The approach here is so that we can see diffraction beats, and this is, if I line scan across here, this is the electron scattering intensity as I move across here. So each one of these peaks here is me looking down a column of titanium atoms, and then all of a sudden we see a hop up here. And this is because platinum has a mass to charge ratio of about 3 times higher than titanium, which means it scatters an electron beam by actually about 9 times higher intensity than titanium does. And so you should see a spike in scattering intensity in the electron beam that goes back down when you get to the next row to identify that you have these single atoms. So this is how the best approach to being able to characterize things by this electron microscopy. If you compare the materials we made using this electrostatic adsorption at very low loadings to those made at 1% or so, you see all sorts of different species here that don't exist here. So just qualitatively looking at the microscopy, it seems this sample on the left has more uniform and mostly isolated structures as compared to the sample on the right. So in my perspective, microscopy provides the best direct evidence that you have single atoms, but it's statistically limited and can be challenging to interpret. So what we've been using a lot is this infrared spectroscopy using probe molecules. We like this because probe molecules absorb everywhere they can possibly absorb. So you're making a statistically relevant measurement. You're probing all of the sites. So for example, if we took those 2 catalysts I showed you on the previous slide, we reduced them at 250 C in hydrogen, and then we absorb CO at room temperature. One shows an extremely sharp stretch, and one shows a broad stretch. The broad stretch tells you that CO is likely sitting in lots of different coordination environments on a surface, and each one of these coordination environments slightly perturbs the CO molecule a little bit differently, and as a result, its vibrational frequency is a little bit different. Also, what you see is the relative position of these bands. This one falls closer to the gas phase CO stretch, which is here, as compared to this one. And that's likely because the platinum that this is absorbing to is cationic. It results in less back bonding or electron donation into CO. And so CO's bond is not stretched as far as it is when maybe you're on a metallic cluster. So we can start to assess uniformity a little bit better by looking at bandwidths. So if you took the full width at half maximum of this sample that we prepared here, where we think we have all single atoms from our microscopy, the bandwidth is a little bit less than 10 inverse centimeters. So what does that tell us? So first you have to consider in the width of a band, an infrared spectra of bound CO to a metal, what the homogeneous broadening should be. So if you just look at the equations for the homogeneous broadening, it's really just based on the lifetime of a vibrationally excited state. So you're absorbing a photon, you're exciting this CO to an excited vibrational state, and then it's going to decay. So the line width should be related to the lifetime that this vibrationally excited state lives. And for CO on most metals, a good guess is about 1 picosecond. And so here you come up with a bandwidth for a homogeneous broadening of about 4 inverse centimeters or so. So we're a little bit higher than that in the samples that we make, but not a ton. This is a really nice paper for an overview of these sorts of broadening things. The second way you can get broadening is inhomogeneous broadening. So if you do a DFT calculation and you put a PTO2 structure on a terrace of anatase TiO2, you stick CO to it, and you calculate the vibrational frequency, you get it up to be about 2121. If instead you make that exact same platinum structure still with the same coordination environment, but now at a step edge, and you stick CO, it's about 2107. So you can see here, this is already broadened. This would be broadened by about 15 inverse centimeters. And if you include the homogeneous broadening of each one of these, you would have a band that's maybe something like 20 inverse centimeters wide, which is clearly much broader than this. And so likely we only have one of these species. So what we always suggest is having less than about 15 inverse centimeters at 300 Kelvin for the CO stretch is a good way to justify that then you should move on and then try to identify the structure of this site. If it's broader than this, likely you have multiple different species, and so structural assignment will need to be challenging. So, next way that you can assess uniformity is you can do a TPD. You can see CO TPD from the sample that had lots of clusters here, shows band positions varying as a function of coverage, and you can see desorption over a really wide temperature window. When CO desorbs from what we think is our sample that has only isolated platinum species in uniform positions, you see the band drop straight down. So no change in the full width half max, no change in the band position. This tells you that the COs, the dipoles of the COs are not interacting. And so these are spatially distinct from each other. So this is the second thing you should convince yourself of before you go and try to make a structural assignment, that you actually have uniform species. So equipped with the idea that all of our spectroscopic characterization told us that we had uniform platinum species on a support, and they were likely all isolated. We then could go say, okay, our vibrational frequency is about 2112. Redhead analysis tells us that our absorption energy of CO is about 0.8 EV. And we can look through a library of different DFT calculations, and these were performed by Gianfranco Paccioni, and look for different coordination environments that give us very similar behaviors. And so what we found is that PTO2 structures, both at a step edge and at a terrace, as I showed on the previous slide, give similar absorption energies and similar vibrational frequencies. So far showing that we're on the right track in identifying our site. We can also look at XAVs, and I won't go into this here. I'll mention this in a couple minutes to show that our structural model here is consistent with a structural model we would derive from an XAVs measurement. And then extremely convincing is if we took this DFT model and we spun it to orient it along a 0-2-1 zone axis, you would find this platinum atom here lines up perfectly with a titanium column underneath it, so a column of titanium atoms. And then if we go do aberration-corrected transmission electron microscopy, what we always find is that the platinum atoms are orienting themselves just above or in line with titanium columns when we're along the same zone axis. So now we have, from infrared, a statistically significant measurement telling us that spectroscopically we have similar species to the DFT calculation. And we have visually evidence that the coordination environment looks the same for the theoretically proposed system compared to this. So from this, we would conclude we're doing a good job of identifying the environment of this site. But we could only justify this because we had uniform species. So that was the next part of the talk on assessing uniformity. I'm going to try to speed up here just so I can finish with time for questions and briefly discuss the structural dynamics. So we identified this coordination environment only for the case where we had pre-treated this sample in hydrogen at 250 C. But you can imagine that this structure may change as you, say, go to more oxidizing conditions or more reducing conditions. So we can probe this using lots of different techniques. Here we've probed this using X-ray absorption spectroscopy. So Zanes measures the transition from a core level state up to unpopulated valence states. And in this case, at the L3 edge, you're measuring mostly transitions into unpopulated D states. And so when you measure Zanes, the absorption cross-section, basically how intense the absorption occurs at the platinum L3 edge, is related to the oxidation state of the metal. If the oxidation state is higher, you have a higher amount of unpopulated states, and so you'll get more absorption. And so what we could see is for our sample that we've been characterizing for the last few slides, the oxidation state of platinum changed as we put it under different environmental conditions. So these were all in situ. And through comparison with standards, we could say that when we pre-oxidize this sample, actually it looks like platinum's in a +4 oxidation state. When we mildly reduce it, as we have been doing at 250C, it looks like it drops to close to a +2 oxidation state, and then even further down to a delta+ oxidation state as we reduce this thing even more harshly. So clearly, the local coordination environment of platinum is evolving. I'm going to skip this just briefly. So we then wanted to see, when we're putting platinum in these different environmental conditions, oxidizing, reducing, harshly reducing, is the change in its oxidation state coming from just breaking local bonds, or is platinum becoming mobile and moving around on the surface? And so what we found is, by doing in situ electron microscopy experiments under these environmental conditions, the platinum itself doesn't seem to move. This bright spot is our platinum species, single atom on a titania lattice. It doesn't seem to move between oxidizing conditions and mildly reducing conditions. But once you put this in hydrogen at high enough temperature, the titania surface itself starts to reconstruct, and so the platinum atom becomes mobile. So any changes in coordination environment that happen between these two states are likely local bond breaking. Any coordination environments here are likely because platinum has become mobile and started moving around. So we went in and we wanted to be able to describe how the coordination environment of platinum was changing as we put it under these different environmental conditions, with remembering that all of the detailed analysis we had done before was on this condition in the middle here. So to do this, you can use XAVs. So XAVs is a part of X-ray absorption fine structure, and this looks at the extended fine structure. And specifically what you're looking at is you have photo-emitted electrons from an absorbing atom that scatter off its neighbor. And when it scatters off its neighbor, it results in periodic oscillations in your X-ray absorption spectra that you can interpret and understand what the local environment is. So here what we did is we took XAV spectrum, and these are complicated wiggles, and I don't expect you to understand the details if you haven't looked at XAVs a lot, for our platinum species under the 3 different environments we had looked at. You can decompose these spectra into the range where you have platinum oxygen coordinations and platinum titanium coordinations. And I'll say here that we had no evidence of direct detectable platinum-platinum coordination. So still is convincing we have these atomically dispersed species. What you can see is as you go from oxidizing to more reducing environment, the amount of platinum oxygen coordination decreases, and the interactions between platinum and titania change as well. So we put this whole story together, and I won't go through every detail, but it's all in these couple of papers here. And what we suggest is, as you move from oxidizing conditions to more reducing conditions, single platinum atoms, their local coordination will adapt. Under oxidizing conditions, platinum wants to dope into a cation vacancy and sit where titanium typically does. It makes 6 bonds to oxygens and some extended bonds to titanium. As you mildly reduce it, it wants to pop out of the surface and sit in a 2+ configuration. And as you reduce it even more harshly, it becomes hydroxylated and becomes mobile and moves around on the support. So this is to tell you that the coordination environment can change of these platinum atoms to these support. And this will happen as a function of the environmental conditions you operate under. We also showed that when you do analysis of the reactivity of these samples in these 3 different coordination environments, they have very different reactivities. So if you do a CO TPD, CO will come off the more oxidized site at much lower temperatures than it does off the more neutral platinum atom that was now just bound to 1 or 2 oxygens and was hydroxylated. So inherently its reactivity is different. If you go measure its catalytic reactivity, again, you see that more neutral species have higher turnover frequencies for CO oxidation than the more oxidic platinum species. And there's all sorts of details here, but what this tells us is that the local coordination environment controls the CO bonding and CO oxidation reactivity. So we can tune this environment and we can make these atomically dispersed species more or less reactive as you tune this, and this is all dictated by the environmental conditions. One note is we could only have analyzed these sorts of structural changes if we knew we were starting with a system that was initially uniform in nature. So it was key that we could produce uniform coordination environment. So then we could use sample average measurements to watch how the system responded as we changed environmental conditions. So I'll leave at the end with just 2 or 3 slides of where we're going further to make more and more complex structures. So you can imagine many ways to increase complexity and functionality. You can imagine putting 2 different sorts of sites next to each other, maybe a rhodium and a rhenium. These sit in very different oxidation states. Maybe you could get one ligand to coordinate here and another to coordinate here, and you could have some bifunctional reactivity. Maybe you could completely change the support landscape and have a support that's multifunctional, alumina, rhenia, all sitting around our atomically dispersed rhodium. Or maybe you could do organic functionalization of the support, so you can do condensation of phosphonic acids, and here you can begin to control the steric environment around these atomically dispersed species, and we're working on all of these different avenues. I'll just show you how we're trying to get to the point where we're making these pairs of sites here. You can do what's called a site-selective deposition that we've tried to develop, where you can take a surface like alumina, can deposit down little bits of rhenia all over. Rhenia has a different point of 0 charge than the underlying alumina, and you can do the same with tungsten as well. And then you can use charged precursors, and you can try to use an electrostatic interaction between the charged precursor with cationic. And these little bits of domains on the surface, which are anionic and have a different point of 0 charge than the underlying alumina, to deposit these right as neighbors. So just to give you a taste of how we've been trying to do this, you can then go and make these materials, and you can use electron microscopy to say, have I made pairs of sites? So you can do aberration corrected in electron microscopy. You can see bright spots, and this is 2 nanometers, so each one of these is a single atom. You can identify pairs, but how would I know if one is rhodium and one is rhenium? Well, rhodium has an atomic number of 45, rhenium has an atomic number of 75, and the scattering cross-section goes as about Z, the atomic number, to about 1.5 or 2. So we would expect a bright scatterer and a darker scatterer. And every time we identified pairs, we had higher scattering intensity, lower scattering intensity. And we could then compose this and say, we didn't perfectly make pair sites because there's other things happening here, but we did make a lot of pair sites, and we can begin to imagine that these will have additional catalytic functionalities than you would have with just the rhodium sitting there by itself. We've taken this a few steps further and gotten better and better at this, and you can see now we can make very nice pair sites of rhodium and tungsten, where we're using the same site-selective deposition of rhodium right next to tungsten, so that we can really push this towards increasing functionality. And in this last case, I won't go through in the detail, but we've pushed to say, understand the behavior of rhodium sitting next to one tungsten atom or one tungsten cation, a 2D tungsten domain, and a 3D tungsten domain. And we've looked at this in the context of a few different chemistries, but particularly ethylene hydroformylation. And what we see is the pair sites. Only when we form one rhodium next to one tungsten, we can see a spike in selectivity. So we can start to say we're looking at beginning to design pairs of sites. And we have evidence that actually the chemistry here is occurring in a bifunctional nature, where ethylene binds here and CO binds here, and they react at the interface. So we can form pairs of sites on purpose that will allow us to have increased functionalities, which we just couldn't get out of clusters or metal particles. So hopefully with that, I can conclude, and I have convinced you of some things, and hopefully you followed some things. I had to go a little fast at the end, that structure-function relationships for these sorts of systems are really nicely developed by quantifying the distribution of active sites in a series of materials and correlating this to some intrinsic catalytic property like turnover frequency. Hopefully I also convince you that not all sites are created equally. If you look in literature, most reports of these single atom catalysts or isolated metal atoms likely have a range of different environments of the metal species in a given sample. And you can tell this just looking at the CO infrared and the inhomogeneous broadening that you would expect. Next is that these metal support interactions are adaptive to environmental conditions. We saw that the coordination environment changed just by changing how much hydrogen or oxygen you put in the atmosphere. And further, that we can really begin to build up uniform and complex structures, and that this is going to be the key to approaching the functionality that, say, for example, organometallic catalysts have. So I didn't really address reaction mechanisms in this at all, but there's a lot to be learned there, and there's a lot to be understood there. So with that, I'll acknowledge lots of funding supports and lots of people that have worked on this over the years. Insu, Joaquin, G among the people here, as well as former group members, Leo and John, Matsubu, Chitra as well. And then the collaborators who have been just fantastic in trying to build up these detailed insights into active site environments. And also thank you again for the introduction. And hopefully I didn't talk too long, and that you all still have a couple questions that I can answer. So thank you.
Thank you so much, Dr. Christopher. That was excellent, and you attracted a lot of interest, so I have some questions for you here. Firstly, what happens with less than 0.5% rhodium loading? Can we increase the fraction of single atom rhodium?
Yeah, so I assume this was relating to the rhodium on TiO2 work. So yeah, that was really just some initial work. That was the first paper we wrote on it. But since then, we went further, and we found that if you go down below about 0.1% of rhodium on most supports that are high surface area, we're typically working on 150 to 200 meters squared per grain surface area. And importantly, you don't reduce this in hydrogen above 200 or 300 degrees Celsius, then you get mostly single atoms. It's not perfect, but along that direction.
That makes sense. Can we use single atom catalysts for petrochemical industry, such as cracking?
Yeah, so actually I know that there is, so I didn't talk about today, and I didn't even really make the relationships, but the relationships should be made, that there is lots of industrial catalysts where precious metals are loaded inside zeolites. Zeolites have really high surface areas, and they also have extremely small cages where you really just can't fit much metal. I know in visiting and talking to folks at Exxon that there is certainly petrochemical conversions where they use fleetingly low loadings of platinum or similar metals in zeolites, such that they want to maintain a cationic characteristics of the metals, and that those are really the active sites. So there are opportunities there. I think a lot of it is done in zeolites, where you also get the advantage of shape selectivity because the zeolite cage imparts some shape selectivity on the chemistry. So I think there's opportunities, but you always have to think first on what the chemistry you're trying to do is. If I'm trying to break carbon-carbon bonds, things like this, maybe it doesn't make sense, and maybe I need acid sites to work with my metal sites to do chemistry. So it's always important to make a link on what chemistry you want to do, and should this active site be able to do that chemistry.
Awesome, thank you. Could you comment on how you've synthesized the rhodium single atom catalysts?
Yeah, so the best way we have to do now, we have 3 different ways. So precursor matters, support matters, and conditions matter. So we try to use either rhodium nitrate under conditions. You cannot go to basic conditions with rhodium nitrate because it will precipitate out and start to form clusters. If you want to go to basic environments to do rhodium deposition, we use this tris ethylene chloride molecule, which I showed, because it's actually stable as a cation. So first thing, we use very low loadings, lower than about 0.3% or so. We optimize pH, which is different for each support because each support has a different point of zero charge. And what you want to create is an electrostatic interaction between your precursor and the support. So with alumina, we go to high pHs, and we use rhodium precursors that are stable as cations because alumina will deprotonate and become negatively charged under high pH conditions. So we wrote about it in a couple different papers, but for each support, it's different, and we play with different precursors to optimize that. But commonality among all of it, lower loadings and not harsh reduction conditions, because that will always make your precious metals mobile.
Thank you. You mentioned how the active site can be at the interface between your support and the metal catalyst. is this true for all single atom catalysts, or are some purely active at the metal site?
This is a good question. I'm just trying to do a counting in my head. Almost every case we've seen involves the support in some ways. If you're doing oxidation chemistries, we've typically done this using reducible metal oxides. And so you actually pull an oxygen out of the support and you do a redox cycle with the support. Hydroxyls on the support can likely participate. I imagine there are cases where entire chemistry can happen on this single atom. So for example, you can do an oxidative addition to convert a rhodium +1 to a rhodium +3 with a hydrogen. And so then you could do hydrogenation chemistry. The always question is there going to be a lower barrier pathway involving hydroxyls from the support? So I imagine there are cases, but even in those cases, I would caution and say, if you put that one atom on a different support, the electron push and pull from that support should be different. And so it still should have an indirect effect on the reactivity.
Awesome, that makes sense. Will the IR resolution affect the spectra of carbon monoxide adsorbed on a 0.025% platinum catalyst?
I don't think any of us have an infrared anymore with worse resolution than 2 or 4 inverse centimeters. So if your resolution is below the homogeneous broadening that comes from just the lifetime of the vibrationally excited state, it should not.
Awesome.
If you look in old literature, you'll find some HRELs, the high resolution electron energy loss spectroscopy, where the bandwidth was much broader, and there you are limited. But with the IRs you can buy routinely now, you have no problem resolving a 10 inverse centimeter band.
Great. And last question, as you increase the dispersity of your catalyst, have you noticed an increase in leaching at all?
Almost everything I showed you today was gas phase chemistry, and all the gas phase chemistry was happening below, let's say, 300 or 400 degrees Celsius. So the leaching there would be volatilization, and I don't think we never noticed any volatilization. If you did go and put these into a solution, we certainly see. So, for example, for tungsten, we definitely see when you have mononuclear tungsten, it can dissolve into solutions easier than crystalline tungsten if you have, say, tungsten oxide on a different oxide like alumina. So there is going to be a difference in leaching properties of the dispersed species compared to metals, because leaching is usually some sort of oxidation reduction cycle that's usually occurring. And if you start with a metallic state versus starting with an oxidized state coordinated to the support, you have a different chemical species. So just like there's this distinct reactivity, there should be distinct leaching as well.
That's great. Thank you so much. I'll just share my concluding slide. So we have here, thank you so much, Dr. Christopher, for your excellent lecture, for all your time presenting it and answering questions. We all really appreciate it.